Hi, everyone. So I'm Ahmed. I'm a fifth-year student in Michigan in the bioinformatics program. And I think this topic is going to be just like dramatically different from some of the topics discussed in the conference. But I'm going to try to show how being like a computational lead in a wet lab, what I'm trying to do to bridge the gap between the translational work and the computational work to derive some insights into pancreatic cancer. So I'll keep going back and forth between the biology and the computational science itself. So I'm working in a lab that's focused mainly on pancreatic cancer. So I'll give you like a little bit of an overview about the pancreatic cancer. It's a pretty dismal disease. It has very low survival rates. So it's like one of the lowest cancers with 5-year survival rate, around like 13%. And the problem that it usually gets diagnosed very late due to having nonspecific disease symptoms. And as you see in the, let me just make that. Yeah. So as you see here, it's usually diagnosed very late, like 53% of the people are usually diagnosed in the late stages. And at that stage, the survival drops dramatically. If you look at the biology, it's hypothesized to originate from a cell type called the Ahssiner cells in the pancreas, which undergoes a process called ADM, and then it gives rise to a preneoplastic lesion called pannins. And pay attention to this. Pannins is like going to be like one of the hallmarks that we're going to be talking about in the talk today. And then in the end, it progresses towards like a full tumor. This is usually happen when there is like a surrounding stroma. So it's not only the tumor cells themselves, there's a surrounding other non-tumor cells that are either helping having like anti or pro-tumorigenic effect. Which is, again, this is like another hallmark for pancreatic cancer. It's not only the cancer cells that are playing a role, there are other cell types that play a role actually in the tumor. One of those cell types is the fibroblasts, and the fibroblasts are heavily studied, yet poorly understood in their pro and anti-tumorigenic effect. And there is a lot of actually clinical trials trying to target the fibroblasts for their role in tumor progression. I assume that most of us are already familiar with single-cell data, but single-cell RNA-seq analysis is one of the methods to be able to decipher the complex tumor microenvironment. And simply what happens in single cell, we obtain the tissue, we isolate like the individual cells, and then we profile the transcriptome of each of the cells. And then we follow with some computational work in order to be able to visualize and analyze this data. So in my lab, we have been doing this for a while, actually before I joined the lab, but they focused mainly on pancreatic cancer samples. So, and the problem is, again, the late diagnosis hampers the idea of being able to obtain normal samples. So we don't really understand the trajectory from how pancreatic cancer develops from normal samples towards being a full tumor. And simply that's because there's no actual indication to obtain a healthy tissue from an individual. Like, you can't just like show up to someone and tell us like, give us a sample from your pancreas. What we've been able to do is we collaborated with an organ donation organization called Gift of Life, and we're able to procure healthy organ donor samples. And the difference in the samples that we obtained from the previously obtained samples from organ donors is we obtained this from brain dead individuals. So the cardiac death individuals, usually the tissue viability after we procure the tissue is so low. But if you look at the graph here, you can see that the tissue viability for individuals who are like brain dead is much higher. So this preserves the molecular data and the tissue structure of the pancreas and helps us understand more about what the normal tissue looks like. So the first step is we had a lot of like single cell samples from the donor samples that we obtained, or like the healthy samples that we obtained, and we started looking at how the microenvironment, what are the differential microenvironment characteristics, and what are the tumor-specific microenvironment shifts. I'm not going to get into details. You can review the paper if you're interested in the different shifts in different cell types, but we see some of the hallmarks. So we see more Ahssiner cells, which are creating the pancreatic juice in the healthy individuals. But then when you get to the tumor, you see enrichment of more immune cells, which is known hallmark for the cancer generally. But we see other cell types, such as the epithelial cells, seems to be having dichotomy. So it seems that we have different shapes and different types of epithelial cells across both tissue types. So the problem that we faced, or the finding that we saw when we analyzed the tissue histologically, is we found that the structure that we know about. So again, we have the acinar cells, we have a transition state called the ADM, and we have the ducts. Again, those are all known to be present in the, or expected to be present in the normal individuals. But what was quite surprising is we found pannins. And if you remember the pannins, I told you those are the penioplastic lesions. Those what precedes usually the tumor. We found those in the normal samples. And it wasn't age specific. We found those in individuals as young as like 20 years old. So that was quite interesting. Again, there's high presence of pannins in healthy individuals, yet the prevalence of pancreatic cancer isn't that high. So the question is, what's going to be the driver? What makes those low-grade pannins present in almost most of us right now become tumorigenic? That's going to be the question. And that's what this type of tissue that we procured helps us build this understanding. So the first step that we wanted to do is we wanted to understand, we have single cell data, right? We wanted to understand, in order to build this trajectory, we want to understand what are the different clusters within our sample that represents those epithelial structures. So we know from the markers and from previous literature, what are the markers for the epithelial cells, like the pan markers for the epithelial cells and the acinar cells, but within this, like, red population, we don't know really how are those different epithelial structures, such as, again, the acinar, the ADM, the ducts, the pannins, until it becomes like full tumorigenic. And the tumor, we have like two stages of the tumor. So we have the well-differentiated tumor when it's still preserving its glandular shape, and then the poorly differentiated when it's losing its glandular shape and it's becoming more fibrotic. So we don't know the transcriptomic signature that differentiates between all of those are buried here, but we don't know which is which. At that point of time, we had the spatial transcriptomics coming up, and it was like voted to be the method of the year in 2020. And there are like different methods to actually analyze or to profile the sample using spatial transcriptomics. One of them is through just like having an ROI, sort of like a bulk RNA-seq for a specific region of interest. And also there's like the Visium, which is the spot level based, and also the single cell or like the near single cell resolution for spatial transcriptomics. Since we knew what we're looking for, we wanted to use the GeoMX platform, because again, we know the histologic phenotype, so we know how different epithelial structures look like, but what we'd like to know is we'd like to understand the transcriptomic profile. So we opted to use the GeoMX platform, in which we chose different regions of interest based on the histological profile that we know of. And after that, I don't know what happened. Can I exit? Seems to be lagging. It froze for a while. Yeah, I think we're here. All right, yeah. So we chose like those different regions of interest, and then we defined their transcriptomic profile using just like simple RNA-seq. And the next step is, again, we wanted to better annotate our single cell data. So what we did is we just like projected those transcriptomic signatures back on our single cell data, and they seem to be, some of them is, so if we look here at the acinar population, it seems to be actually enriching the same regions that we annotated before as like acinar cells. But then we're able to see that certain clusters within our single cell data are actually enriched for distinct transcriptomic profiles that we defined from the spatial data. This helped us. So I mean, until then, 4 years later, we have now more samples. We have like 50 single cell samples. When we integrate all of the data and try to annotate our different cell types, we're able to better annotate different clusters that we couldn't identify before. So specifically those 3 clusters, the ADMs, the ducts, and the full tumor, those are 3 things that we couldn't actually decipher before. We couldn't really annotate before our spatial transcriptomics approach. So that was like the first pass of the data analysis. The natural next question is, as I showed you before, the tumor microenvironment is just a rich microenvironment of interaction between different cell types, right? So we want to understand how this co-evolution happens, how the evolution of the tumor compartment happens, and how the co-evolution in the stromal or the non-tumor compartment happens, from the healthy individuals to being full tumor. Answering this question using the single cell data is quite tricky, especially because even though there are a lot of like cell-cell communication methods that exist that try to impute and infer the cell communication between different cell types, they do not really take into consideration the spatial context. So we don't know which cell type is nearby the other. And the other thing is using the genomics platform is quite tricky because it had a lot of problems with the capture of the stromal compartment. We're able to efficiently capture and have sensitive capture for the epithelial compartment, but for the stromal compartment it was quite tricky. So for that we used the Visium platform. Again, if anybody's not familiar with how Visium works, we have the tissue section, and then there is this like grid in which it has like multiple spots. Each of the spots is around like 50 microns, and there's like distance, and there is voids between the spots. And what happens is within those spots, it captures the transcriptomic or the RNA of the tissue that's underlying those spots. So it's high resolution, it's giving you more data, but the problem is you have more than one cell actually within each of the spots. So it's as if it's like a micro bulk RNA sequences that you have. If you want to showcase this on one of our samples, we see this is like one of our tumor tissues, and if we zoomed in to one of those areas, you can see how the spots look like on our data. Again, each of the spots are overlaying more than one cell. It's not like single cell resolution. So with new data comes new hurdles. So we had 14 different samples. Each sample had actually around 200,000 spots. I'm sorry, 14,000 spots. So we have total of 200,000 spots. So those are our data points. Each of the spots is going to be composed of like thousands or like around like 18,000 genes.
Severe weather alert, or should be okay here.
All right. Should we do anything or?
I don't think so, not for now. It's supposed to be very thundery for the next 45 minutes, so be careful if you go outside. It's a tornado warning. It's a tornado warning?
Yes.
What? I just got a different. That's alarming. Oh no. I don't know. Perhaps you should go. I don't know.
Yeah, it's a severe thunderstorm.
I'm gonna go check with the organizers.
Has been used for the year, so that's what we've got.
I just don't know where we would go.
I can do it. Yeah, we just got this thing.
Should I? All right.
I think warning is the one that means a tornado is actually spotted in the area.
Yes.
So it's
Well, then we just got thunderstorms.
They're turning the hotel for a location to see if they have a particularized place.
I got one for thunder for tornadoes.
Here, is that it?
Yeah.
Yeah, I eventually got the tornado one too. It wasn't the first one I got.
Yeah.
So.
I
didn't get anything.
They were like, he's presenting.
The other session went, yeah, we're asking the hotel staff if there's a place.
Yeah, but they don't.
Yeah,
Oh, you already asked?
yeah, I asked, they're getting information about where in the building you can go.
Nothing ever happens.
I'm sorry,
No, it's fine.
But why don't you go here, then we have to go, we go.
Hi, everyone. Protocol is to stay in the ballroom, close the doors. So we're gonna get everyone in, close the doors, and keep going.
All right.
You are now literally, your attention is literally not counting. So, sorry about that.
It's fine. All right. Yeah. So new data, no problems, apparently. Yeah, so again, at the end of the day, we have 200,000 observations, and within those, we have a lot of different genes that are assayed. So we have like around like 18,000 genes within each spot, right? And the problem is, again, within each spot, it's not one single cell, it's more than one cell. So we have from like 2 to 10 different cells. And what we'd like to learn is, again, how can we learn about the epithelial stromal coevolution from all of this data. So what I've done is I tried to come up with this comprehensive framework of the analysis. So again, we have from the tissue, we have single cell profile. We have around like 40 samplers or so. And when we integrate those samples, we come up with this single cell profile for the pancreas that's composed of tumor cells and normal cells. So hopefully this is going to be our atlas. This is going to be our ground truth for the features that define each cell types. And on the other arm, we obtain 10x Visium samples, again, from donor samples or healthy and PDAC, and also tumor adjacent. And what we do is we do three things in order to be able to characterize the tissue. The first one is cell type deconvolution, which I'm going to get into shortly. Spatially aware clustering, and also we have help from the pathologists in order to better interpret our results. And again, the goal in the end is to characterize the tissue, define different spatial domains, and after that we can start answering our questions. So the first step is the deconvolution. So what we do for the deconvolution? The deconvolution is simply we have each of the spots. We have is simply like a sample, right? And then we integrate this with a single cell atlas, and the hope is we come up with this pie chart, which tells us the fraction of each cell type within our spot. So this is what the deconvolution is doing, and we're doing the same here. We have our single cell atlas that we developed in-house, we annotated and we refined along the years, and we have our samples. Now, I'm not a pathologist, but I've been dealing with the data for like a couple of years now, so I can walk you through some of the things that you should be expecting. Here, like those small islands are acinar cells. This small area is actually tumor. This area is ductal cell, and everything else is fibroblast or like stroma. On the other side, this is healthy pancreas, mostly acinar cells. We have small areas of like ductal areas and stroma. So the deconvolution seems to be working perfectly in the sense of it's estimating the areas where we expected the presence of acinar cells or acinar cells, and tumor, we have like an enrichment for tumor cells, and fibroblasts was expected as well. So we can trust the deconvolution to work. The other part to it is the spatially aware clustering. So again, the clustering, there are a lot of different clustering methods. There are a class of clustering methods that are spatially aware. So they take into consideration the coordinates of the spots. And when we apply those in our method, it seems to be defining different clusters that are that have some geographical arrangement. But again, we start with those, we call them domains. So domain 1, domain 2, we don't annotate them yet. But to annotate them, we use three things. So we use the deconvolution results, and we use the markers within each area, and we use the histology, which is the ground truth. So for the deconvolution, we see the top enriched cell types. So for example, the first two are going to be the acinar, so we call them acinar. This is acinar ADM, so we call them acinar ADM. This is duct. But then we reach this problem of having two different domains. They are transcriptomically distinct, but they have high enrichment of epithelial cells. So we call them initially epithelial 1 and epithelial 2. But then when we look at the histology, we understand epithelial 1 is actually pannins, or like more of like the well-differentiated subtype, and epithelial 2 is more of like the poorly differentiated subtype. And this is actually concordant as well with the markers. So instead of calling those like this, we call them poorly differentiated and well-differentiated tumor areas. And we do the same for the others until we reach like this one as well is interesting. We have enrichment of T cells and B cells. When we look at the histology, this resembles what's known as tertiary lymphoid structures. So those are areas within the histology that are enriched for both T cells and B cells. Again, this highlights the idea, so we call them TLSs. Again, this highlights the idea and the privilege of using spatial data. If we did single cell data, it's going to be two different clusters of T cells and B cells. But here we are capturing that those are actually what's called tertiary lymphoid structures because we capture both of those cell types in the same domain. So this highlights the power of using spatial transcriptomics side by side with the single-cell data. So we end up with this. Representation with this annotation. Those were only 2 samples. In order to visualize all of the samples, we visualized them in a UMAP. And actually, if you look at this differential abundance plot, you can see it recapitulates the same profile that we had in the single-cell data. So now we're trying to answer our first question. So we subset the epithelial cells. You see it performs a beautiful trajectory. The data bigs for trajectory analysis. It starts with the donor panels, or like the healthy preneoplastic lesions, all the way until the late stages of the tumor. So we fit a pseudo-time, we fit the trajectory analysis, we fit a pseudo-time model, and you can see that along the time of the tumor progression here, you have different gene programs that are activated at different time points, which actually matches some of the known programs that are activated in pancreatic cancer. To answer the next question, I'm sorry, I'm going so fast. So to answer the next question is to understand how the fibroblasts remodels as the tumor progresses. So we try to look at the domains where we have enrichment for fibroblasts. And we had fibro1, fibro2, fibro3, and we ignored fibro2 because it seems to be stressed out cells and it's of a lower quality. So we can see that fibro3 is actually localized more around, if you look here at high resolution, they are the type of fibroblasts that are localized around the well-differentiated or the epithelial cells, whether this is in the tumor or in the healthy individuals, while fibro2 is actually localized more with the more progressed type of the tumor, so the poorly differentiated, where there is like more fibrosis and it loses the glandular shape. To have like a more global look, this is a density plot for a whole slide. So again, you can see that fibro3 is around, the fibro3 density is around the high density of glandular structures, while fibro2 is around the areas where we have poorly differentiated tumor. And this was in a tumor sample. This is in a healthy sample. Again, fibro2 seems to be enriched everywhere, but it's also found around the epithelial structures or the glandular structures. So we subsetted those two different domains, and it's lagging again. But the problem is, again, we keep getting back to the same problem of, again, the spot is composed of multiple cell types. So we need to do deconvolution. We need to understand what are the genes in those spots that are coming from the fibroblast. And luckily, there is a method called base prism, in which it's not only getting you the fraction of different cell types within a spot, it's also giving you the gene expression profile, so the fibroblast specific expression within a spot. So it imputes it tries to impute the single cell profile of a whole spatial transcriptomics data. Yeah, it's just lagging. Yeah. So what we do is we aggregate the counts across those different spots, and we generate what we call a pseudo bulk profile to be able to feed it into base prism. And then we deconvolve this pseudo bulk profile. And what we end up with is just another representation for this high dimensional data into a lower dimension. I wouldn't say a lower dimension space. It's, again, here what we're trying to do is we're trying to aggregate the counts in a sample-wise way. So each spot here is a sample which represents the fibroblast expression of a given domain in a given sample. And we changed the name of the annotations. So instead of fibro2, fibro3, we call them now benign epithelial, which are going to be the fibroblast around the early stages of the tumor progression. And then well-differentiated tumor are going to be the fibroblasts in the tumor cells around the well-differentiated state. And then poorly differentiated tumor, it means it's going to be the fibroblast within the tumor. And then we can start doing all the pairwise analyses to understand, again, what are the gene programs that are activated in the fibroblast and in the stroma as the tumor progresses. Yeah, and lastly, just we're feeding in the single cell data. So again, the single cell data is our true ground truth. So we're feeding, again, the single cell data in with this in a sort of a hierarchical deconvolution to see what are the different cell subtypes that are defined from the single cell data in our different domains. So we can see that there are different single cell defined fibroblasts that are enriched in different domains. So yeah, in conclusion, here we're showing that we're utilizing the single-cell anticipation transcriptomics in order to understand the evolution of the epithelial and the stromal compartment. And the future direction is to run cell-cell communication. But at this time, we know for sure that this cell type is close to the tumor or not. So this helps us better with the interpretation and try to define diagnostic and prognostic markers and druggable targets. So I mean, getting back as a closing remark, since I'm working with like computational biologists all the time, and I'm trying to lead the computational science, I would say that all the time you have to start early with them. Try to be in the co-designing of the experiment. Co-designing the experiment. Don't be in the position that you're just getting the data. And understand the biology is messy and it's noisy, so be pragmatic, not perfect. I know that most of the designs in the programming should be anticipating some perfectionism at some point, but again, the biology is very messy and very noisy. And you have to build trust. You have to be able to speak both languages, the biology and the computational science, and build trust with them through letting them know what are you doing and what are the different things that you're doing. So thank you so much. I'd be happy if anyone would like to connect with me.